Okay, everyone could see the lectures on YouTube, especially I think students who missed first two lectures. They're accessible, right? Aditya actually sent the link. So if you click link that, you should be able to actually, you guys should be able to view it. I hope a couple of you have checked.
Yes, sir.
Yes, sir.
It is working well.
They're okay, right?
Yes, sir.
Okay, so because if we need any modifications, we need to be done. So one thing I want to check in that lectures is that because I forgot to switch between the board and PPT. So how did it come in the lectures? Did the board appear whenever I was like? Okay. Okay. I think that will be better. Okay. So because yeah. Okay. Good, good. Okay. So let's say when I share screen, then what will appear in my screen? The okay. So then in your recording, then the board will come. Is it correct, Aditya? Okay. But then it will stay on my cloud only, the account cloud. Oh, okay, on your okay, desktop. You can share it, then I'll put it in the YouTube. Okay. Because those lectures where actually if I'm writing something on the board that's important, that needs to be visible, otherwise it would not make much sense. Okay. So today we are going for class. So this is lecture 3. Okay. So there is a small correction which I want to convey. In the last class, I discussed about cytoskeletal elements. Okay. Cytoskeletal elements inside the cell. So I told about actin fibers, okay, and then microtubules. Okay, so these form really long filamentous elements inside the cell, okay, which partly provides the structure and rigidity to the cell so that it doesn't really become a flattened. Okay, and then I discussed about motor proteins. Okay. Motor proteins which use these cytoskeletal elements to actually do convey cargo from one place to the other. Okay, so I conveyed that both kinesin and dynein. Okay, so walk on dynein walks on microtubules, and then I said that kinesin walks on actin fibers. That's not correct. Actually, both kinesin and dynein and dynein, there are two different motor proteins which use microtubules to convey cargo. Okay, but however, kinesin and dynein, they move in opposite directions. Okay, so kinesin moves from the center of the cell to the periphery, and dynein exactly opposite. Okay, from the periphery to the center of the cell. Actin fibers. So there is another protein called myosin. Okay, so which is very important with respect to muscular tissue, or for example, cardiac tissue, heart tissue. So in which the contraction and relaxation of the entire cell are inside the cell, these fibrous elements called sarcomeres are mainly occurring due to movement of myosin on actin fibers. Okay, so this is one correction I wanted to note down because that was wrongly done. Okay, so today we'll talk about come back to molecules of importance. So let me share my PPT. Okay, so coming back to this table. So this table shows that, so what are the important molecules inside cells, okay? So apart from water, so there are other polymeric molecules or macromolecules. So these are proteins, nucleic acids, polysaccharides, and lipid molecules. Okay, so we'll today start about nucleic acids. Nucleic acids. Okay, so the well-known nucleic acids, of course, DNA and RNA. Okay, for the reasons that DNA acts as a genetic material, so transfers information or blueprint of information. What is required for the making of a cell, or tissue, or entire organism, and how that information is actually passed from one organism to another organism. And RNA also plays an important role. Okay. So we'll discuss about the details. Okay. So what are nucleic acids? So they have certain elements. Okay, so they have what is called sugar. I'll show the structures in PowerPoint. So there is a sugar. There is a 5-membered sugar, pentose, right? Okay, so sugar is attached to a nitrogenous element called a base. Again, I'll show you structures. And then sugar is also attached to a phosphate. So this is basically a common architecture of nucleotides.
Sir, I'm unable to see the board, sir.
So you're not able to see the board. So in your now you see?
We can see.
Yes, sir.
Yes, sir, we can see that now.
Now I think even when I share slides, you should be able to toggle between PPT and my image. So you need to know that how to do that.
Okay, sir. Okay.
Okay.
Sir.
So this is a base structure of a. Sorry again. Someone is asking a question. Okay. So this is the base structure of a nucleotide. Okay. So if we don't have a phosphate, a sugar plus okay. So if only sugar plus base is called nucleoside. Okay. So basically, this is without phosphate. This is very important demarcation. Without phosphate. Okay, so this is one thing. I'll show you now the structures in my PPT. Okay. So on the board, you see basically base structure. You have a sugar, and then sugar is attached to a base, which is a nitrogenous element, and sugar is also attached to phosphate. So all three components together, it's called nucleotide. And then if you don't have a phosphate, if it is only sugar plus base, it's called nucleoside. So this is very important demarcation because these words are actually used if you read the research papers. They say nucleotide and nucleoside. Okay, so everyone sees my PPT now?
Yes, sir. Yes, sir.
Yes, sir.
Okay, so now the sugar is a 5-membered ring containing 1 oxygen and 4 carbons. And then there are a lot of hydroxyl groups. Okay, so this is 5-membered ring labeled carbon 1, carbon 2, carbon 3, carbon 4, and typically use a prime to differentiate between these numbers between sugar and base. Okay, so 1 prime, 2 prime, 3 prime, 4 prime, and 5 prime there is CH2 attached to 4th carbon. So this is sugar. So this is pentose sugar. Okay. Now the base is attached at the carbon 1 prime. Okay. So again, I'll discuss about what are these bases. And at the 5 prime carbon, this is a phosphate group is attached. Okay, PO4 2-. So there is one hydroxyl group which is labeled at C2 prime carbon. Okay, if that hydroxyl group is present, then it's basically ribonucleic acid. Okay, if that hydroxyl group is replaced by a hydrogen, then you have what is deoxyribonucleic acid. Okay, so DNA and RNA. So now phosphate is fixed and the sugar ring is fixed separately for deoxy and oxy. Only the base is different now. Okay, so there are different bases, 2 different groups. So one is okay, so bases you have 2 groups: purines and pyrimidines. So these are 2 different types of bases. Okay, so purines are basically what we call macrocyclic, but 2 rings, and pyrimidines contain single ring. So there are 5 bases. Okay, so adenine, R single letter A. Is it set up or no? Everything is fine. Thank you. Guanine, G, cytosine, C. So these 3 are common for both DNA or RNA. So there is a 4th piece, thymidine or T for DNA and uracil. Okay, or U, so for RNA. So this is one base which is different between DNA and RNA. Okay, so the structures are there on the right side of this slide in PPT. So adenine, 2 ring membered purine. Guanine is also 2 membered ring purine. Cytosine, thymine, and uracil are single ring pyrimidines. And the difference between thymine and uracil is basically a methyl group, okay, present on carbon number 5. Okay, so the numbering is on the left side. So pyrimidine, so nitrogen 1 to 2nd carbon, 3rd again nitrogen, 4th carbon, 5th carbon, and similarly for purines. Okay, 1, 2, 3, 4, 5, 6, 7, 8, 9. Okay, between the thymine and uracil, the main difference is there is a methyl group which is present on carbon number 5. Okay. So this is basically the basic architecture of nucleotides and nucleosides. So nucleotide contains sugar, base, phosphate. Nucleoside contains only sugar and phosphate. Okay, so these are called nucleotides. And so now you use these nucleotides to make the macromolecules which we call DNA or so
may I ask a question?
Just a minute.
Sure.
So DNA and RNA are okay polymers of nucleotides are polynucleotides. Okay, so we'll see that how exactly the connection is between the nucleotides to make a polynucleotide. Mitradi?
Yes, so uracil and thymine, they differ only in a methyl group.
Correct.
So is there any chemical reason for that? That why while forming DNA you needed that extra methyl group?
Yes, I think why they came into existence most probably through evolution to induce the complexity. But chemically, how they are introduced is basically when they are synthesized. So you have two enzymes, okay? So which make DNA and RNA. So let's say if you have a DNA sequence to begin with. So let's say you are making another copy of DNA. So then you have DNA polymerase. Okay. So DNA polymerase recognizes only thymines when they copy from one strand to another to make a copy. But however, let's say you're making RNA, okay? So then you have from DNA, so then you have RNA polymerase. Okay. So RNA polymerase is a different enzyme than DNA polymerase, though of course both bind DNA, but when they make the complementary polymer chain based on a parent chain, so they recognize different elements. So looks like RNA polymerase recognizes only uracil. Somehow that methyl group on that position is important. Finally, how these things really occur is that recognition between a coming nucleotide and the enzyme. So that enzyme recognizes that based on the complementarity, then only I think that's I think two-way argument. One is through evolution to increase that you want to separate RNA structures from DNA, both sequence and structure.
Okay.
Second is that you have two different enzymes, so the way they recognize.
Sir, right now it's difficult. I am not able to toggle between the board and the slide. I mean, I was able to before. So, is there any setting issue?
No, I have not done it. Setting. Aditya, what?
Okay. No, still the board is showing minimized. I mean, I am not able to maximize the board part because earlier I was able to. Today itself.
Well, I don't know. Just check pin video. If you do.
Yeah. Now it's enabled, I guess.
Or I don't know. There are a couple of options. One is pin video. There is another is spotlight video. I don't know the difference, but one of them should work.
Yeah, right now it's possible. Yeah.
Okay. So it's good to actually practice these structures, okay, as a chemistry student, so that if you do once or twice, then at least you'll be able to appreciate the differences when you see these structures again, let's say in research papers or elsewhere. Okay. So then we know the basic nucleotide structure, okay, or the framework. So the DNA, deoxyribonucleic acid or ribonucleic acids are nothing but polymers of these nucleotides. Okay. So in the case of DNA, you have AGTC. In the case of RNA, you have AUGC. So these are the 4 nucleotides which can be used to make polymers. Okay. But again, when you make these polymers, what's important is directionality. Okay, so what do I mean by directionality? For example, let's say if I take a DNA sequence, which is consisting of, let's say, a nucleotide followed by G, followed by C, followed by T, then let's say again G, let's say A. Okay, so this is some example sequence where my first nucleotide is adenine, followed by guanine, then cytosine, thymine, etc. Okay, so this directionality is important. So similarly, if you take for RNA, again you can write analogous sequence. So let's say you read again A, and then you have let's say G, then you have C. So RNA doesn't have thymines. In the place of T, let's say you have G. Okay. So then how do you represent this? First, okay, let's see how to make, let's say, this sequence of polymer and where the connectivity. Okay, that's very important. So on the slide, you see on the left side, again, simple architecture. You have the sugar, 5-membered sugar, where the base is attached to carbon 1 and the phosphate is attached to carbon 5. Okay, so this is called, for example, this nucleotide is base is adenosine and phosphate is at 5 prime. Okay, C5 prime. So it's called adenosine monophosphate or AMP in small form. Okay, and this is specifically phosphate is at this 5 prime. So sometimes it is called 5 prime AMP. Okay, so now let's say if you have to attach another, okay, so to begin with for on the board and showing it, let's say first is A, nucleotide you already have. Okay, so let's say now you have to attach G, okay, guanosine nucleotide. So where do you attach to this on AMP? Okay, so where do you attach? So you attach at the C4 prime, so C3 prime, there is a hydroxyl group. So you replace that hydroxyl group, okay, so with phosphate of the coming nucleotide, that is guanosine, okay, and so you just eliminate water from, I guess, okay. No. Okay, so you basically instead of that OH, so you attach to that phosphate. So on the right side, you see on the right side picture, the first one is the DNA and the second one is RNA. So at the DNA, you see the second nucleotide is thymine coming. The phosphate is attached to 3 prime carbon. Okay, and the third nucleotide is guanosine. Okay. So now, so this sequence is important, and this sequence is called 5 prime to 3 prime, both in DNA and RNA. Okay, why it is 5 prime? 5 prime is because, so you want to say on the sugar, okay? So the first phosphate is actually attached to the 5 prime carbon, so that's beginning. So then 3 prime is the second nucleotide is attached, and then to this 3 prime, the next base is attached. Next nucleotides 5 prime is attached. So that's why it's called 5 prime to 3 prime. So when we write this sequence AGCEGA, for example, here, so the sequence is automatically meant that it's 5 prime to 3 prime. So this is different from, let's say, if I have to just reverse the sequence AG. Okay, I'm writing from the backwards AGTCGA. Okay, so these two are different from each other because in this case 5 prime and 3 prime, the sequences are different. So this is one very important notation one needs to really remember. So that's why explicitly sometimes you write 5 prime so that you say that the sequence is actually running, because it's very common when you work with DNA sequences, you work with what's called complementary sequence, which I'm going to come. So you exactly reverse the sequence. Okay, well, just simply writing the reverse doesn't mean that it's 5 prime to 3 prime, it's 3 prime to 5 prime. So either you explicitly mention 5 prime to 3 prime or 3 prime 5 prime, or just if you don't write any labels like 5 prime or 3 prime, it's implicit that it is 5 prime to 3 prime. Okay, so this is how the connectivity of nucleotides, okay, in a polynucleotide. For example, here it is APG in DNA or use GC in RNA. Someone has a question?
Sir, Shreyas here.
Yeah.
Yeah. So I have a question here. Like in case of RNA, when the phosphodiester bond is being formed, why does the 5 prime position go and bond specifically to the 3 prime position and not to the 2 prime position when both of the recepting hydroxy groups are secondary hydroxy groups?
Yeah, so that's correct. Again, see, most of the questions in biology are through evolution rather than why they really occur in different pathways. Okay, so maybe initial reactions, okay, for example, might have occurred at both positions, you know. So in the case of RNA you're talking about, because in the case of DNA you already have a deoxy, so there is no
Yeah.
okay.
There's no question of any selectivity in case of DNA.
Exactly. For example, of RNA, where there is a hydroxyl group at 2' carbon, so you're talking about why not my phosphate could have on reached there or not.
Yes.
My answer would be most probably through evolution, somehow maybe both have tried, but somehow this worked out to be
Worked out. Okay.
this. And yeah, this brings to me another important point. So as Shreyas points out, okay, so there is a hydroxyl group at 2' carbon in RNA, which is absent in DNA, so that's why we call deox. Okay. So this hydroxyl group again is an important element in RNA. Okay, if you have, let's say, very alkaline conditions, okay, so there is a base catalyzed reaction, okay, so which actually reacts this hydroxyl group now can attack the phosphate on 3' to cleave this phosphodiester bond, and then RNA is fragmented into individual nucleotides. Okay, so where actually there is a cyclization between C 2' and 3'. So this is one separate thing which doesn't really occur in DNA. So DNA is a robust molecule, very stable. So this doesn't really, this kind of cleavage doesn't really occur in our DNA, but RNA is susceptible to this hydrolysis. Okay, so as a chemist, you would be interested in basically looking at that reaction. So that's actually, it's a very well-known thing, base catalyzed cleavage of RNA. Okay. So, so now we have seen the sequence, rough sequence of connectivity.
Excuse me, sir. Excuse me.
Yes.
So, sir, can you please repeat that why the reverse sequence is not happening of DNA? The 5' to 3' and not if we reverse that sequence. So, can you please repeat that? I could not get it.
Okay. So, what I said is that in RNA, you have, let's say, so this is 5 membered ring, right? So this is a 2' carbon. Okay. So where there is OH. Okay. So this OH is absent in the DNA. So that's why it's called deox. So now let's say when you have phospho linkage. Okay. So this oxygen, okay, can now attack. Okay, so this basically base catalyzed. Okay, so if you have alkaline medium, okay, so which can actually activate this, and then this can actually attack phosphodiester bond, and then there is a cleavage of this. Okay, so this, because of this cleavage, now the polynucleotide chain can be fragmented into individual nucleotides in RNA. This doesn't really occur in DNA because this OH is, which is responsible for this reaction, is absent. Okay, so that's why sometimes you say that RNA is not a very stable molecule. Okay, so in conditions like DNA is actually. So that's one difference just I wanted to highlight. Okay, so this is the connectivity, right? Connectivity of nucleotides in DNA and RNA. But what is the structure? What is the structure of this polymer? Okay, so because if you see there are a lot of single bonds. There are single bonds on the phosphodiester bond. There is OP is a single bond, PO is a single bond, or that oxygen connecting to carbons are single bonds. Okay, and then so because almost all are single bonds, so you know that as a chemist, the rotation can really occur on these single bonds, and then you have huge conformation freedom. Right? So then, what might be the structure of these polynucleotides as they exist? Okay, so people have tried to look at the structure of these nucleotides, and of course, a lot of work has been done on DNA because DNA could be really purified in reduced quantities, and it forms basically kind of fibers. So in the 1940s. So basically, even before that, a lot of work has been going on, and X-ray diffraction has just come up. 1905 is basically first, I think, Roentgen X-rays, okay, 1905, the first Nobel Prize, basically began looking at diffraction of molecules. So in 1940s, I think this guy called Erwin Sharp, okay, of course a lot of others. Okay, so he made some observations based on his research as well as colleagues and others. So what they looked at it is when they looked at basically the composition of nucleotides, okay, especially DNA. Okay, so because we could really purify DNA from, let's say, organisms, okay, the bacterium or any other species, it's very easy to purify. If you walk into any molecular biology lab, it's right now, of course, it's much easier. But even earlier days, it was much easier to purify DNA than protein. So based on the DNA material purified from different species, he made actually some observations. Okay, so this DNA composition, of course, by then we already know that DNA contains only A, G, T, C nucleotides. Okay, so you could really measure the content of A, G, T, C, okay, and stoichiometry, their relative stoichiometry. So what is observations he has made? One, he made observations that the composition of DNA varies from species to species. Okay, so if you take one species from other species to another species, okay, means different, composition is different. But however, interestingly, if you make DNA from one species, or let's say one organism, okay, composition is constant. Okay, across ages, DNA composition is constant. It is age independent. Okay, age independent means that, so if you take, let's say, from mouse, okay, so if you take just take a tissue, okay, from tissue you purify DNA. The composition in terms of how much A and G or T or C are present. Okay, if you measure literally in terms of concentration, that's independent. It's independent of what the mouse might be eating. Okay, independent of its environment. Okay, independent of whether it is old or as it gets old. And even more important. Okay, so it's across species, tissues, independent, same in all tissues. Okay, so if you take from DNA from, let's say, lung tissue versus muscle tissue, the composition is exactly same within species. Okay, same in all tissues within a species. Or within an organism, you can call. Okay, that's very interesting information for you. Okay, and most striking is that if you purify DNA from a cell, remarkable observation is that concentration of A is similar to T. Means that you can really correlate higher the amount of A, higher the amount of T. And concentration of G is exactly same as. Okay? So this is a remarkable observation that concentration of A in this DNA is same as T. Concentration of G is same as C. But concentration of A and G varies different species.
Sir, can I ask a question?
Yes.
Sir, if the DNA is same in all tissues, in a particular organism.
Yeah.
So then what makes them have different function? I mean,
No, DNA doesn't have different function. DNA is blueprint. Copy is the same, but how that information is read, okay, and interpreted, okay, in terms of what proteins constitute different tissues is different. DNA is exactly the same.
even the sequence of bases.
Yes, so basically you have the same nucleus. Nucleus is the same. DNA, chromosomes, for example, in us, chromosomes are same. Okay, but the thing is that, let's say lung cell will have different sets of proteins versus muscle cell. Okay, brain cell will have different sets of proteins, but the DNA in all these cells is exactly the same one. Okay, so these sometimes are called Chargaff roles. Okay, so even in the absence of any structure, so this information is actually kind of starting point to understanding what might be the structure. Mind you, this is 40s. Okay, so we don't have much structural studies. Okay, not even start. But parallelly, what I'm showing on this PPT slide, so work of Rosalind Franklin, Maurice Wilkins, very important one. Okay, so both are pioneers in X-ray diffraction. So looking at basically DNA structure. Okay, so what they are looking is at basically purified DNA fibers. Just under diffraction. Okay, do we get any periodicity? Is there any periodicity or not? Are there any periodic elements? Stop. Okay, so combining with Chargaff rules, where the important observation of concentration of E is A is always equivalent to T, concentration of D is equivalent to C, but A and G themselves can vary. Okay, and this X-ray diffraction study from Rosalind, the picture I'm showing on the PPT on the right side. So this is basically kind of well known picture. This is the first diffraction pattern of DNA. Okay, so where they say that, I'm not really expert on X-ray diffraction, but what they say is that you see the central X dots, which are actually constituting as X at the center, says that there is a helical structure. Okay, so that's all the hint. There's nothing more than that. There is a helical structure. Okay, so there is a helical structure, and then you can actually quantify this. This helical structure has two periodicities. Apparently, you can actually quantify this image to numbers. Okay, so one is 3.4 angstroms periodicity. Means something is actually repeating at 3.4 angstroms. But another periodicity is at 34 angstroms. Okay, exactly 10 times bigger. So that's the only information, but beautiful fine information coming from this diffraction. Okay. Now, if you combine this information with this, so the DNA has, okay, this concentration of pyrimidine is exactly same as adenine, and concentration of G is same as this. Okay.
Sir, how the separation, the 3.4 angstrom and the 34 angstrom was decoded from the crystallographic structure?
That's what I'm telling. I'm not really expert on X-ray diffraction, unfortunately. But they say that from this, see, you see the spots?
Yes, sir. Yes, sir.
And you see the, for example, maybe directly how far are these spots? and at what angles they are. This is kind of X, right? So you can really again think of what is the angle between these two arms, okay, and what is the spots difference, okay, how spatially they are, okay.
Okay, sir.
You can use this information to get to these two numbers which I'm showing on the board, two periodicities. But other than that, I'm not really equipped with how to exactly extract this from this. Okay, again, the figure legend says that the heavy bands at the left and right arise from the recurring basis. Okay, so
Okay, sir. Okay, sir.
yeah, I'm sorry for that because I'm not really experienced.
Okay, sir.
So,
I have a question on sample preparation by Rosalind Franklin. So for obtaining such a clear case space image,
yes,
you will need some good periodicity happening.
yes.
So did they use somehow did they crystallize the DNA and then used it for imaging?
No. I don't think they have crystallized anything. I think they have, most probably they have purified directly from species. I believe, I'm not sure. So you can actually purify DNA a lot, something like half-timers DNA. You can actually buy literally almost in milligrams to grams quantities from sigma. Means that its purification is so well robust. Purification of proteins is nightmare, but purification of DNA is relatively easy. Okay, so that I can really vouch for that. So let's say you have cells. Basically, you break open cells. Basically, DNA and RNA can be separated very easily. Okay, from proteins because DNA has much larger molecules. Okay,
Yes.
I think this is the first chapter in the Leninger. So there actually one of the figures shows that how to actually separate different components of cell. Okay, for example, in centrifugation, you can actually do what is called ultracentrifugation, where you can do what is called sugar gradient. So let's say you lyse the cells, means break open all the contents, and you can actually separate DNA very easily because DNA is much larger molecule than anything else inside the cell. Okay, and then which actually get precipitated much easily. So you can separate any other molecules, okay, from DNA. I think that's what they might have done, but I don't think they have really crystallized. I don't know. I don't think it's like, I think these are just, they say fibers. So because though you don't really require, I guess, even crystals, I guess, just highly purified DNA itself will give you this. Okay. So what you are talking about in crystals, how these fans might be, let's say double-stranded DNA structure.
Yeah, because if the orientations are slightly different, the image in the case space will be all messed up.
Good point. That's true. You might get hazy spots, but still the periodicity is, see, you're looking at, okay, different scales. Okay. Let's say even if you have double-stranded DNA, much larger, but clumped together like a rope. Okay?
Yes.
The periodicities which we are looking at angstrom level.
Yes.
They will be present. Okay?
They will be there.
Yeah, it's not that you need to really stack all these ropes again like a bundle. Might not actually required. Okay. So I think that's not okay. So now then comes Watson and Crick. Of course, this is most famous story. Okay, most celebrated story, and people even consider even today this is the most important finding of biology. Okay, not even just modern biology, entire biology so far. Because what Watson and Crick have done is that they have actually read this information very carefully. The chaga fruits, the concentration of A is equivalent to T, G is exactly same as C, and these periodicities. Okay, so now they have to really model. Okay, simply just like jigsaw puzzle, model that what are the structural organizations. You have to incorporate helical structure with this kind of periodicity with that composition. Okay, how exactly it will gives rise to structures. So then in 1953, famous Nature paper, mind you, this is only 1.5 pages. I'll actually forward you that article. Very short, just 1.5 pages in Nature. They propose, okay, they haven't done any new experiment, believe me. They have just taken the information from the Charlie Ruffles. They have actually used the Rosalind Franklin's this diffraction image, okay? Just based on that, they propose the structure. So what they are saying is that, so if you, so now we know polynucleotides. So polynucleotides have phosphodiester bond, sugars, and bases. Phosphodiester bonds, okay, are basically phosphates, highly negatively charged. So you have phosphate, negatively charged. Okay, so negatively charged. Bases, okay, hydrophobic. Okay, sugar. So you have a bunch of groups which form hydrogen bonds. Okay. So this is the information they have. Okay, at their end. For phosphates have highly negatively charged. So you can't really put together a lot of charges clashing with each other. Okay. So parallelly, actually, Pauling is also proposed a structure which turned out to be wrong. Okay, but which actually exists in under specific conditions. I will talk about in the next class. So Pauling thought that phosphates are actually clumping together, stabilized by, let's say, ions, and the bases are actually stacking out. But the Watson and Crick, what they said is that, okay, bases are hydrophobic, so they need to be protected from hydrophilic or aqueous environment. Okay, so they need to be exposed as less as possible. Sugar has
one question.
Yes. Who is this?
So how did they come to the conclusion that bases are hydrophobic? Because ultimately within the DNA structure, they're forming hydrogen bonds with each other.
No.
So
this is information before the structure. Okay, before, let's say, Watson and Crick structure. So if you see the bases, you have, and let me go back to a couple of slides. Okay, so if you see this slide, adenine. Okay, you see all these adenine. Okay, so if you see, let's say this is a planar structure. Okay, so you have a lot of this pi cloud, right? Okay, a lot of pi cloud. So this pi cloud doesn't want to really interact with water. Of course, you have NH2 and NH, only these two groups which can donate hydrogens, but you have a lot of surface area which is hydrophobic. So you need to actually protect this hydrophobic surface, okay, because it can only form van der Waal interactions or maybe kind of very weak dipole form. Okay.
So what can we comment about the, is the sugar part, what do we call it? Should we say it hydrophobic or hydrophilic? I mean,
no, sugars are soluble, so these are hydrophilic because they have OH groups which can be hydrogen bonded very easily. Solubility is very high.
Okay, thank you.
Okay, so but however, so phosphates you have two negative charges, O minus, bases hydrophobic. Okay, and sugar just has maybe in DNA we are talking about, so OH has a hydrogen bond capacity. Okay, so then how to come up with this sequence? So then what they thought is that again at the same time they have information that DNA is two strands. Okay, DNA is two strands. Okay, so two strands of nucleotides. That information is also there. But now they have to really put together this helical structure. Obviously, you can wrap around these two strands because again the sizes of those two strands are exactly same. Okay, and overall content of the both strands is this, not the single strand. A concentration same as T, G concentration same as C is from both strands nucleotides. But within the same strand, this doesn't really hold. Okay, again they know this. Basically, you have to apply this rule A, T, and GC across strands. And then now they are thinking about, okay, you have two equal sized strands, different sequences, but they have to form helix. Okay, so first they thought that, okay, why not we put these two strands in helix? Okay, so if they put helix, then they have to really think about now where you have to keep the functional groups. So Watson and Crick thought exactly opposite to what Pauling thought. Pauling thought that, okay, phosphates should be inside and they form, let's say, helical structures. Watson and Crick said that no, because you have highly negative charge, they will be huge revolutions. That's not a stable structure. So let's put phosphates outside and bases inside, because you know exactly the connector. So the moment they put that, so this is the structure they came up with. Okay, so phosphates outside, good, because then the solubility will be very high. Hydrophobic bases, okay, so how to actually separate them from water? They made actually stack. Okay, so stack means that, so one base, okay, as you move from the bases from 5' to 3' or 3' to 5', each base one above is actually stacking, okay, in the same strand. So this stacking actually makes them interact through van der Waals interactions and also partly dipole-dipole. Now, because of that interactions, they are out of water. That's good, okay, because it's really high. So second thing is that then how to connect these bases with the neighboring strands. Okay, so strand 1 and strand 2. Then they came up with, okay, so the moment you put the stacks on each strand, now the stack will need to interact, okay, what do you call it, head on, okay, head on, and then they came up with this hydrogen bond. Okay, so automatically it's forming into this hydrogen bond forming structure. Okay, so once they thought, okay, and then of course they have to really think about hydrogen bond means that which base to which base, and then comes handy that this concentration principle that A equals to T and G equals to C. Automatically they're saying that, okay, you have exactly the same number of A and exactly the same number of C. So somehow this equivalence means that A and T are interacting. Okay, A and T are interacting and G and C are interacting, because automatically somehow nature conserved this concentration. So automatically when they applied to this, then they were thinking that what might be the interactions, and this comes beautifully automatically this hydrogen bond. So adenine and thymine, these are single, no, adenine is purine and the guanine is purine. So, and thymine and cytosine. So beautifully, adenine and thymine could give rise to two hydrogen bonds, okay? So sharing between them. And cytosine and guanine could exactly provide 3 hydrogen bonds, okay? So that they can satisfy between them. This beautifully kind of just locked in with these additional constraints they have to really. Okay, and then sugar, of course, this OH is basically outside. Okay, so we'll come back to this 2' OH because that gives rise to RNA different structures. Okay, so now then quantitative information. So Rosalind and Morris found that this periodicities, right? Now then you have to really apply this periodicity, 3.4 angstroms and 34 angstroms. And beautifully, when they put this helical structure, they could really see the 3.4 is basically base stack difference. Means the height of the base stack as you move along the DNA helix. So the rise is 3.4 from one base to another base in the stack, and 34 is basically so repetition of structures roughly about 10 nucleotides. As you move along the one of the strands, okay. So and this gives rise to a famous double helix. Okay, so DNA double helix structure, as we know today. Okay, so this is only a proposal, but final refinements really led to exact numbers and etc. Okay, so for example, so if you see this structure, they say 36 angstroms for 10 base pairs and 3.4 for base pair, right? But Rosalind's numbers are 34 angstroms, not 36 angstroms. Okay, so somehow, so if you have DNA in fibers, somehow it's basically small differences in the structure because these are fused fibers. Gives rise to rather, let's say if you have isolated double-stranded DNA. If you have double-stranded DNA which is isolated in water, has slightly different structure than DNA in fibers because these are much. Okay.
So I want. So
Yes.
how did they come to the conclusion that DNA is two-stranded and that two hydrogen bonded? So can you please repeat that?
So those experiments can be done very easily. So even before you get to the structure, you can really do, for example, let's say heat denaturation. You know, so you can actually separate. Even if you take, let's say, large DNA fiber, you can fragment it, okay, by different methods, and then if you heat it, you know, so you could actually separate these strands. So then you can actually isolate these strands and then say that, okay, somehow one strand sequence is different from the other strand, but you have two different sequences running in the sample. Okay, so that's what I actually alluded. So you have, so when you isolated these molecules, okay, so they have different sequences. One has one, okay, strand one and strand two, let's say. Strand one and strand two, which can be separated very easily if you heat. Okay, for example, let's say it's 95 degrees Celsius. So they get separated. Okay, so there are methods even by then to actually chop into fragments and then get the sequences. Okay, so then they found out that from all those experiments that any the DNA in DNA, let's say purified DNA, you have actually two sequences running, okay, parallelly. You know, but however, only when you combine the content of A and G gives rise to this concentration in proteins. That's actually how. Okay. Okay, so this is basically, okay, so this on the right side, what you see is that cartoon picture. Extreme right, you see the space filling model. Okay, so if you see in that space filling model, clearly it's a right-handed double helix. Okay, so it's a right-handed helix. Okay, and it has, you can see two grooves. One is called minor groove and major groove. Minor groove and major groove. Okay, so two grooves, you can really see that major groove is basically a lot of space as you walk around the helix, but minor groove you have a little less space. Okay, so this is a famous double stranded DNA. And
Sir, I have a question.
Yeah, and you can see the diameter is about 20 angstroms or 2 nanometers. Okay, so it was possible because of all this progress in DNA research. Because the content, for example, the polymers are made up of only 4 elements, AGTC, unless unlike proteins, which will come back later, much more complex. Second, they could give highly purified DNA could be purified to high concentration and purity, and then luckily very simple DNA diffraction patterns. So could give rise to basically DNA double helix structure. But you will see that protein structures, of course, started very innovatively. Max Perut's hemoglobin, myoglobin studies. But however, again, protein structures took really long time. Okay, there is a question.
So I want to ask that Watson and Crick gives us the model of that structure. and Pauling also gives a model of that structure.
Correct.
But how actually, what is actual structure? How scientists know and how they got Nobel? The experimental have proved as there, because from X-ray we cannot say that the phosphate is outside or phosphate is inside. Only from X-ray. So how they got Nobel? Linear Pauling is wrong.
Nobel Prize is given because very okay. So not only that, so these are actually I forward that paper. So they not only propose this structure, but this right away explains a lot of unknowns by them. Okay. So for example, let's say how a copy of another DNA or RNA made based on DNA. Okay. So they could now really see that. So right away, if you have this structure, imagine, let's say, if there is a protein binds, this is exactly walking around this double stranded and making complementary strand. How genetic information is actually transformed, okay, or read up from these molecules. So it would, they even propose that, for example, let's say they even proposed codons by now, okay, so codons and how another complementary strand could be made. Imagine, let's say now you unwind one of the strand, and then if you put an enzyme, if it is reading from one end to the other end, either it could be 5' to 3' or 3' to 5'. So you could really write down the sequence of letters. If that sequence of letters corresponding to your RNA, okay, a lot of information is there, all this information is there, but there's no connection. You know, how exactly this is translated is not really known. But the structure clearly provides that if you unwind the structure from one end, and then if you read that information, okay, based by nucleotide by nucleotide, this way, you could really translate that into a copy of another DNA molecule or a copy of another RNA molecule. That really provided right away this structure. Because without this structure, you have no way to say that how exactly this information is read out. We know that the information is actually, so there is something called central dogma. So for example, DNA gives rise to RNA, gives rise to protein, right? Okay. So without this DNA double-handed structure, we could really not know how exactly all this information. We know that every cell has the same copy of DNA, and we know yet different proteins are present. Okay, so we know that DNA is responsible somehow relating information, but how exactly could not be proposed in the absence of any structure. But this first proposal right away actually explained a lot of, at least gave insights to things which we know. For example, replication of DNA, or making another copy of DNA, or even kind of translating this information into protein. Okay, so but they didn't have to really do experiments, but let's say the insight itself really heartbreaking, you know. So that's groundbreaking, I would say. I think that's what. So I'll forward that paper. You should really read it. Just 1.5 pages. Right away, let me do that. I said it's first page, second page, not even half actually. I would say it's barely crossing 1 page. Okay. And you see the beginning sentence. Okay, we can't really write papers anymore. We wish to suggest a structure for the salt of deoxyribonucleic acid DNA. See, they're not even saying that we are confident. They're saying we suggest. Okay, that's it. That really shows that kind of insights which are required. Means that itself is pathbreaking. Okay. And of course, they say that a structure. Sorry, I'm not sharing. Not sure. Okay, now you should be able to see. So this is basically first page, and the second page is hardly just 1 or 2 lines. Okay, everyone sees my slide, right?
Yes, sir.
Okay, good. So you see the first line. Okay, the title: Molecular Structure of Nucleic Acids, the structure of DNA. We wish to suggest a structure for the salt of DNA. Okay, they are not saying that this is a structure. They are just suggesting. And you see the second paragraph, first line. A structure of nucleic acid has already been proposed by Colling and Corey. They kindly made their manuscript available to us in advance of publication. Means that was not even published. Their model consists of 3 intertwined chains with phosphates near the fiber axis. Okay, and the bases on the outside. That's exactly opposite what actually they are thinking. In our opinion, this structure is unsatisfactory for 2 reasons. Imagine this is basically the audacity of the authors. One, they're taking manuscript from friends that's not even published. Second, they're saying is that already it's not published, but it's even wrong. Amazing, no? We can't really do these kind of tricks now. So their model consists of 3 intertwined chains with these phosphates near the fiber axis and bases on the outside. In our opinion, this structure is wrong for reasons. We believe that the material which gives the X-ray diagrams in this is the salt, not the free acid. Without the acidic hydrogen atoms, it's not clear what forces would really hold the structure together, especially as the negatively charged phosphates near the axis will repel each other. Some of the van der Waals distance appear to be too small. And then, of course, they come up with this 2-span. The figure is purely diagrammatic. See, imagine what they're writing in the figure. The figure is purely diagrammatic. The 2 ribbons symbolize the 2 phosphate sugar chains and the horizontal rods of the base. Okay, but somewhere they say that the elaborated details will be published elsewhere. Okay, you should read basically in 5 minutes. You can actually read this. Okay, so but you should see the last couple of sentences. Okay, so it has not escaped the right side second paragraph from bottom. It has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism of the genetic material. Okay, so that's all they're saying, means that this structure has a way to explain how the strands might be copied, how actually you can make another strand using this. Okay, that's all they're saying. Okay, fuller details of structure, including the conditions assuming in building it together with a set of coordinates for the atoms, will be published elsewhere. Okay, and nevertheless, this paper is cited well and not really the paper where they have given the details. Okay, so I think this is the most cited paper, I guess, in biology. Okay, so really, so coming back to, yes, this is double-stranded DNA. So this is called bDNA. Okay, if I remember correctly, this is called bDNA. Okay. Okay. So, but Pauling is not really wrong in some sense that some structures actually where phosphates will be pointed inside. Okay. So yeah. Directionality of okay. So directionality comes automatically. Okay. So what they're saying is that okay, 2 oppositely running strands. Okay. So okay, that's a good question. Okay, so 2 strands are in one. Okay, so they thought about whether these strands should be parallel or anti-parallel. Okay. Okay. So now, so they know 2 strands exist. Their sequences are different. Whether they could be in parallel or anti-parallel. So parallel, I mean that 5' to 3' strand should be paired with another 5' to 3' because we know the order. What is 5' 3'? So is it pairing is parallel or it should be anti-parallel. Okay, so when they try to put the parallel, okay, so somehow they have to again come back to this matching of equivalence. Somehow, how many A's exactly is same as how many T's, how many G's exactly same as how many C's. So when they try to do these two combinations, the anti-parallel automatically actually matched that this sequence, not only sequence that A is pairing with the T, G is pairing with C, but the concentration of A and it somehow explains that why you need to have exactly same number of T's and the same number of C's for same number of G's. Okay, so that's how actually they decide. So it's not that initially they know that right away it is anti-parallel. So means that, so finally, of course, the right structure is, so one strand runs from 5' to 3', the other strand, okay, runs exact opposite. So 3' to 5'. Okay, so they are oppositely going strands. So it doesn't really matter in the end, but you have to really write that one strand clearly when you write the sequence, the top strand typically you write 5'. I think that should answer your question. So they thought about both parallel and anti-parallel, but the parallel didn't really work.
So can I ask a question?
Yeah.
So what is the, so DNA is rightly circular. So what is the advantage of right-hand helix? Specifically, why DNA does not form left-hand helix?
No,
so is there any advantage?
No, next class I will show you the structures where left-handed helices are also possible. Okay, so right now it's basically the connectivity. You know how the nucleotide architecture. Okay, so the phosphate at the 5', okay, and 3' connecting to the next nucleotide on the sugar, giving rise to this helicity. But imagine if the connectivity is not 5' and 3' on the sugar, 5-membered sugar. So, coming back to this question. So basically, this connectivity. Okay. So this connectivity is giving rise to your structure. Okay. So second thing is that. So this is 1', 2', 3', 4', and 5'. Okay. So second is that. So the sugar, how many atoms are there? There are 5 atoms in the sugar. Okay. So there is something called buffer structure. So we call that the sugar ring is puckered, means that so not all 5 atoms are in the same plane. Okay, so at a time either any 4 atoms are in the plane. So typically either 3' or I think 4' they go out of plane. Okay, so that's one reason why you have a stable structure only in the right-handed helix but not left. Second is where the connectivity. The connectivity is at 3' and 4'. Imagine, let's say this connectivity had been on 2', then you might have maybe totally different structures. So this connectivities will restrict you how the bases can be freely stacked. Okay, so that gives rise to the helical structure. Helicity is actually imposed by connectivity.
Okay, thank you.
Sir, I have a question. In the DNA topological structure, you are getting the base pairing between purines and pyrimidines. But why do we not expect the base pairing between purines and purines or pyrimidines or pyrimidines? Is there any Watson and Crick proposes the model depending upon the experimental data they have? But why we not expect a purine base pairing or pyrimidine base pairing?
No, but if you see, okay, let me see the structure where actually I'm doing. So this structure you see on the, let's say adenine pairs with thymine because adenine, see total number of hydrogen bonds, whether given or taken, is basically two atoms. Okay, so the nitrogen and then one hydrogen on the nitrogen. Okay, so let's say if you have to
Sir,
have
are you showing some slide? Because the slide
yes,
is
I'm
not
sharing
visible.
some
The paper is
slides.
visible.
I'm
It's not
sharing.
shown.
The paper is visible. The slide is not visible.
Okay, but I always okay. Let me share the slides again. Oh, somehow it doesn't switch. I didn't realize that. Okay, so now you see this base pair structure. 80.
Yes, sir. Yes, sir.
Yes, yes.
So if you see the hydrogen bonds, so between adenine and thymine, two hydrogen and bonds are possible because each one is giving one. Each one is giving one donor, and each one is actually have one acceptor. Okay, but if you see G and C,
Yes, sir.
there are total 3 bonds. Now let's say if you try to pair G to T or A to C, so then you will not have completely satisfied the hydrogen bond requirements. Okay, that's one reason. Second thing is that so this complementary nature comes because how they are arranged in the parallel anti-parallel strands. Okay. So let's say GC, GT and AC, when you have circumstances like there are something called triple helix, you know. So if you have, there are third strand can come and form actually hydrogen bond.
Okay, sir.
Then you will have totally different architecture. And next class, I'm going to discuss about, this is a canonical double stranded structure. But it's not that all DNA structures are double stranded. If you don't have a complementary strand, imagine, let's say if you don't have entirely complementary strand, and if you have only single strand is available, so then the sequence within the strand can fold back and then form interesting structures. So one of the structures is formed is what are called quadruplexes. Means that there are 4 strands, 4 bases come together and form beautiful hydrogen bond network. Okay, and this is possible towards what are called telomere structures. So if you see the nucleus, it's a chromatin, but at the edges of the chromosomes, there are some G-rich sequences up there. So these are entirely G-rich, means that you don't have enough complementary C. So then all the G's clump together and form beautiful hydrogen bonded base pairs. G forms hydrogen bonds with G, you know. But 4 such guanine bases come together and form what's called G tetra. I'll show you structures next week.
Okay.
So next week we'll talk about non-canonical DNA and RNA structures. Okay, and why RNA doesn't really form double helix. Okay?
Sir, here we are seeing that when the base pairing is occurring between purine and pyrimidines, purines are slightly bigger than pyrimidines. Let's say purines have 2 rings are fused. So is there any steric reasons or any structural features which allows the only purine pyrimidine base, or is it not the purine or pyrimidine base pairing?
No, it's not the steric at all. Basically, how perfectly you can actually satisfy hydrogen bonds. You know, G can form hydrogen bond with thymine, no problem. But the thing is that, so then it's not optimized. Let's put it like that. It's not optimized. Okay.
Okay, sir.
If you have a choice, then of course the canonical form is the most stable. But when you don't have a choice, these are the examples I'll give you next class, where you don't have a complementary strand. You don't have a complementary strand at all. So then, of course, now you want to really optimize the long, let's say, single DNA strand. How do you optimize the structure? Because you don't have complementary strand. Then there is no double helix. Then you have to come up with structures which might be more suitable for that. And these are called telomeres. Telomeres has ends of the chromosomes. You know, there is a Nobel Prize actually, you know, for the recognition, for the identification of telomeres, especially, you know, how the chromosomes are actually shrinking during evolution, and how actually our cells, when we age, imagine one cell is actually copied to another and another and another. As you actually replicate the cells, somehow you have to actually finish the copying of the information. Imagine one chromosome needs to be completely copied. So you can't really say that, okay, I copied 99%, okay, let's leave 1%. This is called shrinking of chromosomes. Somehow there are telomeres. These are really allow us somehow say that protecting the hands, okay, and which have non-canonical structures. And these are actually very important. Okay, so I'll show you non-canonical DNA structures.
Okay, sir. Okay, sir.
Class today, but read this DNA paper. You know, very short, and I mean, you really wonder that. Okay, so this is how exactly the research is done those days. Yeah, because something, you know, so we can't really write papers like this anymore. No one really even. Well, abstracts themselves like page long these days, you know, and also you can't really write this sentences like this. Okay, thank you. My friends actually provided a manuscript which is not published yet, but however, we feel that they're already wrong. You know, it's ironic, but that's what really happened in the case of DNA. Okay, so see you on Friday. Thank you. Bye bye.
Bye, sir.
Thank you, sir.
Hello, sir.
Thank you, sir.
Can I have a last question?
Sir, I have a question.
Me too, sir.
Okay. You go
You,
first.
Prithvish, go.
Prithvish, go.
Okay. Sir, you mentioned, I was stuck in that part only, that every kind of cell within a particular organism has the same DNA. I am not an expert about this, but I have heard that cells replicate by something called central dogma. I mean, is it right?
No, central dogma is what actually I mentioned. So the DNA information is translated to RNA.
Yeah.
So this then makes protein. That's central dogma.
Okay. What I am trying to say is that, let's say you have a heart cell and a lung cell. So they both have same DNA, so same set of codons and all. So shouldn't they create same kind of protein?
No, yeah. So there are. So this is. It's only DNA is only the what is the message? Okay, but however, how it is read depends on the circumstances, cell conditions, intracellular conditions, lot of other factors. Okay, why, though all genes do exist in every cell, why only certain genes have actually expressed? Okay.
So by expression, you means that let's say DNA have a particular sequence. So in a heart cell, certain might not get read. Certain information might not be translated. On while in lung cell, some information.
Yes. So this DNA to RNA. Okay. Not all genes are expressed in all cells.
Okay.
So this is spatio-temporal, means that it's time dependent, which genes are expressed, which are dependent on the conditions inside the cell as well as outside are important, which genes are translated. Okay. So there is something called differentiation. So during the development, as life starts with single cell, when it divides to two, more or less both cells are identical. Okay, but as it divides more and more, okay, so now cells need to differentiate into different organs. Okay, so they already separate into, okay, this cell wants to become a muscle cell, this cell wants to become a brain cell. Already the information, okay, is encoded there that, okay, few cells need to be heart, few cells need to be brain. So that information is passed at the stage of, let's say, a few hundreds of cells. Okay, until that point, all cells are identical. But then slowly this bifurcation occurs. This is called differentiation. Okay, means that the cells are destined to become only certain tissue types. Okay, so once they are differentiated, it's not going back. The differentiated muscle cell versus differentiated heart cell will have different information already inside the cell. So you have the same nucleus, but however, the molecular content, okay, already existing small molecules versus large molecules in terms of proteins, the content is already different. This content itself now further dictates, okay, what genes need to be now expressed in these cells. Okay, so that information is rather then, okay, passed down during the differentiation, will control in future course of these cells. And then though you have, every cell has now the same DNA information, however, only few genes are expressed because it's basically every time you have to tell the nucleus what needs to be expressed, okay, what needs to be copied. So that information goes into the nucleus depending on cell types. Okay.
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 So stem cells doesn't have any kind of.
No.
Okay.
Yeah. So stem cells are basically the beginner cells. That's what I'm saying. During the development, initial stages, all cells are identical. These you can call stem cells because they have not really decided what they want to become.
Okay.
Okay, but down the lane, so slowly dictates that, okay, it's not that you need to have a rich environment, you need to have few cells need to function as brain, few cells needs to function as, let's say, eye.
Okay.
Okay, so then already that bifurcation occurs. Okay, so these are called differentiated cells. These are not stem cells anymore. Okay, but you already have stem cell pool, okay, in your bone marrow. Yes. Okay, so where actually there are circulating cells, sometimes cord cells, human cord cells. You know, when the babies are born, their cord has actually stem cells. So if you actually save those cells, you know, you can actually freeze it, and then that's the amazing thing about cells. You can really freeze it, and then you can make them available even after decades. In principle, actually, they are really frozen and protected. The stem cells.
Okay.
But the moment the environmental cues, okay, the outside molecular environment dictates to differentiate cells. Okay.
Okay, sir. Okay. Thank you.
Sir, I have a question.
Yes.
My first question is, sir, can you repeat that what is the work of motor proteins?
Initially what I said.
Yes, yes.
Okay, so in the last class, I discussed about cytoskeletal elements. There are three classes. Okay, one is actin filaments, second is microtubules, and third one is intermediate filaments. Then I discussed about microtubules are used by motor proteins called kinesin and dynein. So last class I wrongly told that kinesins actually use actin filaments. No, kinesins actually use microtubules, but there is a different molecular motor called myosin, which uses actin filaments to walk around. And this is a very important phenomenon, especially in cardiac cells, where actually cells continuously being contracted or stretched. So during this phenomena, myosin actually play a very, very important role. But nevertheless, it's present in non-muscular cells as well, because you have actin stress fibers. So myosin actually is binds there. So that's the correction I wanted to make. Okay.
Sir, another question.
Yeah.
Sir, we know that X-ray can damage cancer cells. So, sir, in the Rosalind-Franklin experiment, X-ray can damage the DNA also. So, how they experiment that with X-ray diffraction? Because X-ray has a chance of breaking DNA.
That's a good question. So then how do all the crystallographers actually get their studies?
Yes, I also puzzled that because I written that X-ray can, nowadays X-ray can damage DNA for cancer cells. So I thought that how they do.
No, I think it's yeah, good question.
DNA cannot absorb X-ray. DNA can absorb UV light only.
So okay. Exposure time. And what you have in DNA is basically these organic bases, you know. So these are actually aromatic, right? I mean, you have huge pipelines. So these are the where actually radiation like UV can be absorbed. Okay, so that's actually one of the ways actually we detect DNA is by their absorption. Okay, but you have to really see in the X-ray. Okay, what wavelength they are using. Okay, so if that wavelength doesn't really excite directly any of these chromophores, then the energy is not absorbed directly. Okay, so but cells have a lot of other molecules. Okay, a lot of other metal ions, coordination complexes. Okay, so where actually you have material where they absorb, maybe not exactly DNA, but DNA UV actually damages DNA. So one of the ways is that thymidine. So thymidine, I think dimers they really form, and then they can actually further cleave the DNA back. Okay, so that is one of the ways actually DNA is damaged by UV, but not really X-ray. Okay, and as Aditya points out again, the dose, how much you are using, and how long actually your exposure is really important. Okay.
Okay. Thank you, sir.
Okay, I think that's all for now.
Yeah,
And
sir.
yeah, Subra.
Sir, I have heard that the mutation of the DNA occurs. So even for by environmental exposure or for any kind of different reason. So you said that composition is constant. I mean the age independent. So if can the DNA mutation occur?
So definitely, that's a good question. Yes, the mutations can really occur, but our cells are very clever. There is something called DNA repair mechanism. There are molecules which are dedicated, okay, to make sure the information which is copied from one cell to another cell during the cell division is faithful, means that the correct replication occurs, and these molecules which do make mistakes sometimes during the copying or editing. Copying, there might be mistakes occur, but there is something like editing, just like you edit your Microsoft document to see the correctness after you type a paragraph. There are molecules, DNA repair enzymes, they actually correct this. So they can also mitigate any UV-induced damage or any other damage. Okay, there are DNA damage response, dedicated molecules inside the cell, which actually correct this information. Good question.
So then DNA mutation does not occur at all. I mean,
mutations do occur, and they will be corrected inside the cell. Okay.
By themselves, it will get corrected.
Yes, because there are ways actually to know that it's not only okay. So one enzyme just continuously goes and copy DNA information. Okay, another enzyme come back and quickly checks whether the copied DNA scan is correct or not. Okay, how it's correct? If it is not correct, so the base pair mismatch occurs. Okay, let's say A is complementary with T, but instead of T, a G is replaced. Then A and G in the double-stranded DNA doesn't form a perfect base pair. Okay, so this match can be recognized by another one, and then it says that no. Okay, so this actually there is a bulge, DNA bulge is here. So you go back and correct. So again, another polymerase comes, and there is editing mechanism. So it can be corrected. Inside the cells, yes, we do. But outside the cells, if it happens, it happens. We don't know. For example, like in vitro studies, like you copy DNA in the test tube, then if a mistake occurs, there is no correction mechanism because you don't have all that cellular machinery. But inside the cells, yes, it can be corrected.
Thank you.
Okay. Okay, that's all. I think we'll take questions if there are any next class. Okay, see you on Friday. Okay, bye-bye.
Bye, sir.
Bye.
Bye.
Thank you. Bye.
Bye, sir.
Bye, sir.